Today's lesson is Unit 1, Topic 1.3, Macromolecules. Macromolecules are very large organic molecules. Macro means large. They are all composed of smaller parts that are joined together by very strong covalent bonds. There are 4 types of macromolecules that we're going to learn about: carbohydrates, lipids, proteins, and nucleic acids. But first, we need to talk about how we build and break down macromolecules. Dehydration synthesis is the name of the process in which covalent bonds are created to build macromolecules by removing a water molecule. So dehydrate means to remove water, and synthesize means to build. So in order to build a macromolecule, we must remove water. So if you look in the example here, we have 2 glucose molecules. And if you notice, at the end of the glucose molecules is a functional group. I'm going to give you a second to see if you can remember which one it is. An OH is a hydroxyl. So you have a hydroxyl group and a hydroxyl group at the end here. And what happens is one of those hydroxyl groups and one hydrogen from the other hydroxyl group come together and make a water molecule that is removed. And then that oxygen is left as the linkage between the 2 original glucose molecules. And we have a water molecule as a waste product. So that is dehydration synthesis. That's how we build. To break down those covalent bonds, we're going to do the opposite. We're going to add water. So we're going to hydrate. So hydro means to add water, and to lyse something means to break it. So we're going to add water to break those bonds. So again, here we have the opposite reaction. We have our 2 glucose molecule here, our maltose, that has our oxygen linked together, our covalent bond here. We're going to add water to it, and those hydroxyl groups are going to return, and we have 2 individual glucose molecules. So dehydrate to build and hydrate to break down. So hydrolysis and dehydration synthesis. A monomer is another vocab word that you need to know. A monomer is a smaller unit, a subunit of a macromolecule that is linked together to form the large organic compound. For each of the macromolecules that we're going to talk about, we will specifically talk about what the monomer of that particular molecule is. And the polymer is just the name for any macromolecule that is made of more than one monomer. So our first macromolecule that we're going to talk about are carbohydrates. Carbohydrates are always made of a ratio of 1 carbon to 2 hydrogens to 1 oxygen. So it can be any combination of those. The one that we talk about the most, if you recall, is glucose, which is C6H12O6, which does follow that 1 to 2 to 1 ratio. Carbohydrates are found in sugars and starches. Their main function is energy. We will talk about the different forms of that energy in a few minutes. They are also for some structural components in different organisms. The monomer is a monosaccharide, that means 1. And then the polymers can be disaccharides, which is 2, and poly, which means many. Monosaccharides. Glucose is the most common one, right? Mono means 1. Ribose is another example of a monosaccharide. This is the sugar that's found in RNA, ribonucleic acid. Fructose is the monosaccharide found in fruit. And then galactose is another one that we won't talk about very often, but it actually has the same chemical formula as glucose. So it is C6H12O6, but in a different arrangement. Can you remember what that's called when it's the same chemical formula but a different arrangement? That is an isomer. So dehydration synthesis, when we build these molecules, right, we dehydrate to build, results in a particular type of covalent bond called a glycosidic linkage. So when you take two glucose molecules, for example, or two monosaccharides, and when we're linking them together, it's called a glycosidic linkage. So you do need to know that. That is the covalent bond that forms after dehydration synthesis. And these are some of the common results of that. So disaccharides, di means two. So we have maltose sugar, that is a glucose plus a glucose molecule. Sucrose, which is table sugar, the sugar that you put in your coffee, for example, or you bake a cake with, is a glucose molecule plus a fructose molecule. And then lactose, which is the sugar in milk, is made of a glucose plus a galactose. Again, you don't have to memorize all of these. These are just common examples that you should be somewhat familiar with the names of them. And down here, you can see the process of dehydration synthesis one more time. We have a glucose and a fructose. We have our two hydroxyl groups. That water is removed. We've created now, this right here is our glycosidic linkage that forms our polymer, or our disaccharide in this case, where we have two sugars that are linked together to make a disaccharide. Polysaccharides, remember poly means many. These are larger molecules. These are made of long chains of individual monosaccharides, or the individual monomer of a carbohydrate that are linked together. One example is starch. Starch specifically has the names amylose, which is what we'll refer to it as, and amylopectin. This is how sugar is stored in plants. Plants use starch. Think of a potato. A potato is a starchy vegetable. That is the storage unit for a plant. They are made of chains of glucose molecules. They are all made of alpha glucoses, so that means that they are easily broken apart because it's all the same glucose. They're all lined up in the same orientation, and they can be easily broken down by enzymes, which is why starch is very digestible to people and to other animals as well. We're going to talk about some polysaccharides that are not very digestible in a couple minutes. Glycogen is another type of polysaccharide, again for energy storage. This time it's for animals. Glycogen is for storage of energy in animals. Again, this time it's branches of alkoglucose molecules. Animals store this in their muscle cells and in their liver. This is where we have insulin and glucagon involved in the formation and the breaking apart of glycogen. So insulin is the hormone that triggers the formation of glycogen after we eat. It removes that excess sugar that's floating around in our bloodstream, and it stores that energy for us for later. When we need it later, we rely on glucagon, that is the other enzyme involved. It is a hormone that triggers the breakdown of glycogen, and then it releases sugar back into the bloodstream for us to be able to use. So insulin triggers the formation of glycogen after we eat. And then if we haven't eaten in a while, but we get up to do a little bit of exercise, we're going to use glucagon as the hormone that reverses that, takes the glycogen, and now makes it readily available sugar in our bloodstream. People with diabetes, both forms of diabetes, have issues with their insulin production. They might make too much of it. They might not make enough of it. This process right here causes some issues for them. And then we have polysaccharides that are used for structure. Cellulose is the most abundant polysaccharide for structure. This is found in plants. This creates that tough stem material of a plant. It also holds a plant upright. This is made of branches of beta glucose, not alpha glucose, right? The starch was made of alpha glucose. This is made of beta glucose, and the shape of that molecule, the arrangement is slightly different, where now we have some of these glycosidic linkages that are formed in this direction and some are in this direction, and they keep alternating like this. This is what makes cellulose undigestible to most animals, because the enzyme that tries to go through and break these particular bonds here when we consume this has a really hard time with the fact that the molecule, those glycosidic linkages, that covalent bond, is facing different directions. Remember, structure is really important in biology. So the structure of this molecule makes it almost impossible for that enzyme to break it down. It can break down one side, but it can't break down the other side of the molecule. So this makes it a challenge for people to digest it and other animals. As a result, it makes it what's called soluble fiber. This molecule in a digestive tract actually stimulates the digestive tract to secrete mucus, which actually moves feces through the intestines. If you didn't have soluble fiber, if you didn't eat some of this material, sometimes it's called roughage is what the nickname for this is in more common terms, it actually stimulates your digestive tract to move things through. Otherwise, everything would just kind of get blocked up in there, and that would not be a good thing. In other animals, they have a polysaccharide for structure called chitin. It is mostly found in the exoskeleton of arthropods, which are insects and crustaceans, as seen over here in this diagram. And it also is found in fungi, like mushrooms and stuff, and it makes the cell wall up of those. Again, these are for structure, so it's a very thick, tough material that is designed to give some kind of shape to whatever organism it is. So that is part 1 of macromolecules.